welcome everybody. It's a pleasure to present our work on novel strategies for stroke therapy based on the targeting of mitochondrial dysfunction and ER stress signaling pathways. The work I'd like to present this morning includes the efforts of 3 laboratories at Florida Atlantic University. Those laboratories are my own, that of Dr. John Wu, and that of Dr. Rui Tao. And with this combined approach of 3 different laboratories, we find that this combined expertise has been very valuable for production of the appropriate models and testing of the signaling pathways and drugs that we're interested in. An overview of the talk will discuss the neuroprotective functions, firstly of the amino acid taurine, a discussion of the drug DTC-MESO, which is a disulfiram metabolite, as well as taurine as agents for potential stroke treatments. And as a third area, multi-drug strategies for stroke therapy. And these include a triple drug strategy involving a growth factor that may mobilize stem cells but also elicit neuroprotection, a preconditioning agent is a separate drug, and a third drug, the one I mentioned, DTC-MESO, this disulfiram metabolite. So those 3 drugs we've administered simultaneously. I'll present some data on those with individual administration and also combined administration. So I'd like to introduce taurine as one of the most abundant amino acids, and it shows remarkable neuroprotective functions. It's known to play a role in various tissue functions in a variety of tissues, and is very potent neuroprotectant. In some tissues, it's involved in osmoregulation. It has antioxidative functions in neurons and other cell types, and it's also involved in neurotransmission. I'll present briefly some of the background data on these functions. Firstly, taurine has been recognized as being important for neuronal migration, and studies by Sturman demonstrated in a taurine-deficient animal there were defects in development of neurons in cerebellum and in the visual cortex, and the abnormal cerebellum showed defects in granule cell migration. Similar things were found in the visual cortex, and there was these could be corrected if taurine was given an appropriate time window. I'll discuss its role as a neurotransmitter modulator, as a neuroprotective agent that may modify both apoptotic functions and ER stress functions, its importance in maintaining calcium homeostasis. And its role in osmoregulation is well recognized in a variety of cell types, including kidney cells, but I won't focus on this function. This data illustrates that taurine can reverse or prevent glutamate-induced neuronal toxicity. And so you see here measurement of lactate dehydrogenase in neuronal cells in culture with glutamate eliciting high-level cell death and taurine reversing this, the cell death. So taurine plus glutamate gives less cell death. We investigated some of the contributing factors to this. This is a confocal analysis using a calcium-sensitive dye, and you see that glutamate elicits a high-level increase in intracellular calcium. This can be washed out. Taurine has no such effects. But with taurine plus glutamate, there's a diminished alteration in intracellular calcium. And then you see that the cells are still viable because when you treat them with glutamate again, you get a massive increase in intracellular calcium. So this is an example of calcium elevation that's intracellular, that free intracellular calcium that's reflecting the glutamate toxicity that can be reversed by taurine. And this just reflects the same kind of experiment with taurine administration and then glutamate treatment, but no elevation in intracellular calcium. So the taurine can prevent the glutamate-induced elevation of intracellular calcium. And to try and analyze the channels that were important and also the source of intracellular calcium, we have this experiment testing intracellular storage of calcium. And what you find is that taurine has no effect on the mobilization, but if you administer taurine and then glutamate, there's no major change in intracellular calcium in D and E. But when you treat with pertussis toxin, you see that intracellular stores of calcium are released. So taurine is actually preventing release of calcium from intracellular stores. And if we then look at some of the pathways of cell death, initially apoptosis, you see that taurine plays a significant role in preventing the glutamate-induced elevation of caspase 3 activity, seen in lane 2 here. So 2 concentrations of taurine diminish the elevated calcium caspase activity associated with apoptosis. In looking at the apoptotic pathways, we've shown that taurine can increase BCL2 or prevent the induction of BAX by glutamate. So you see that here the ratio of BAX to BCL2 is augmented with glutamate treatment, and this is reversed by taurine treatment. So in this case, the BAX-BCL2 ratio reflecting a level of apoptosis. So just to summarize a few of the contributors to the glutamate-induced apoptosis and the sites of action of taurine. Taurine is capable of diminishing depolarization associated with glutamate and as a consequence preventing the influx of calcium via NMDA receptor. Taurine also acts to prevent mitochondrial elicited apoptosis and also the calpain contribution, because calpain is a contributor to apoptosis in neuronal cells in the neuronal culture model we're employing. Taurine also prevents the formation of BAX BCL2 heterodimers in apoptotic function and also prevents cytochrome C release. It finally contributes to the role of cytochrome C in activating the caspase cascade in downstream apoptosis. Now I'd like to turn to the role of taurine in ER stress to introduce some of the endoplasmic reticulum stress pathways. And these we've investigated in depth in our stroke models. So endoplasmic reticulum stress is very important, both in stroke and other neurodegenerative diseases. It's known to be important in Alzheimer's and Parkinson's, and can play a role in diabetic-induced alterations in brain function. Common to many of these disorders is the accumulation of misfolded proteins in the endoplasmic reticulum that play a role in both elevation of cellular ROS, but also signaling through endoplasmic reticulum stress pathways. Initially, one has an induction of an unfolded protein response, which I'll talk about shortly, but under severe conditions of severe stress, ER stress ensues with activation of one or more of three major pathways. In this study by Oida et al., it was shown that the ER stress marker, GRP78, co-localized with NUN, so that neuronal ER stress was indeed elicited, in this case in a gerbil model of ischemia, and shown here at 1 day after ischemia. So that's an example where ER stress is indeed elicited in neurons under ischemic conditions of brain ischemia or models of stroke. So I'm going to describe the three major ER stress pathways. I just want to show you some of the abbreviations that apply to these pathways. So a marker of ER stress is the GRP78 protein. One of the major pathways is PERK or PKR-like endoplasmic reticulum kinase. And a second pathway is the ATF6 pathway, and a third pathway is the IRE1 pathway. And then other abbreviations apply to EIF2-alpha, eukaryotic translation initiation factor, ATF, activating transcription factor 4, XBP1, Xbox binding protein, and CHOP, which is one of the downstream activators of transcription downstream of the ER stress pathways. So here you see the major pathways: PERK, signaling to EIF2-alpha, modifying translation and signaling to ATF4, and changes in GRP78. ATF6 pathway, which signaling here to CHOP. CHOP is a regulator of the relative contributions of BAX and BCL2 and pushes the pathway towards BAX and or BIM and pro-death components. In this case, BIM is shown, BIM being a pro-apoptotic component eliciting cell death. And the third pathway is IRE1, signaling through XBP1 to CHOP. Additional pathway that we will mention as I go along is a caspase 12 that's associated with the ER, tends to be associated with the ER membrane, and itself can elicit downstream cell death processes. So it's certainly very interesting that under conditions of initial stress, one can elicit these responses through these pathways, IRE1, ATF6, and PERK, that show protective functions that are part of an unfolded protein response. But under more severe stress, one sees that there's activation of CHOP and hence pro-apoptotic components, BIM, and downstream cell death. So we first tested if, in our cell culture models, if glutamate would induce ER stress. And using different concentrations of glutamate, we found that there was elevation of ER stress markers, GRP78, CHOP, the caspase 12. also EIF2 alpha, ATF6, the cleaved form that's active, and then the IRE1 pathway. So these are embryonic neurons in culture that form differentiated cellular processes in culture and cell cultures that respond to glutamate treatment. Wanted to test if taurine could protect against ER stress induced by glutamate. And you see here that that glutamate does result in a decrease of viability, shown here as ATP release. So with glutamate, you have a lot of cell death, and then the addition of taurine is highly protective, as shown here, when it added to glutamate-treated cultures. So taurine inhibits glutamate-induced ER stress, and you see here glutamate-induced elevation of CHOP reversed by taurine, likewise with BIM, similar with caspase 12, similar with GRP 78. And these graphs just illustrate a quantification of Western blots that have measured these changes. Again, CHOP, BIM, caspase 12, GRP 78. Interestingly, shown here, taurine plus glutamate does not reverse EIF2 alpha elevation or ATF4. But it does reverse ATF6 as shown here, or IRE1 shown here. So in summary, those treatments with glutamate blocked the elevation through the ATF6 pathway and the IRE1 pathway, but not the PERK pathway. So the third pathway, the PERK pathway, was not protected by taurine, just the ATF6 and IRE1 pathways. We've done similar experiments with hypoxia reoxygenation and found very similar results with taurine inhibiting ATF6 and IRE1 pathways, but not the PERK pathway. And I won't show those today because in this session because of time constraints. But I'd like to move to some of our work on stroke using a in vivo stroke model. And we've employed the rat model of middle cerebral artery occlusion. It's well known that stroke is a major cause of death in the United States. And we've focused on ischemic stroke, which constitutes 85% of strokes, not on hemorrhagic stroke. Interestingly, there is excessive release of certain neurotransmitters in stroke, including excitotoxicity by glutamate and aspartate neurotransmitters, and a role for NMDA receptors, as well as influx of calcium sodium chloride ions and efflux of potassium ions. Ultimately, a loss of cellular integrity. And this, again, shown here, we have a role for the NMDA receptor, excessive depolarization, and a major role for calcium release from intracellular stores. Ultimately, one kind of apoptotic and necrotic cell death. First drug that we specifically analyzed using our stroke model was DT-CMESO, a disulfiram metabolite. It's interesting that this drug is the active metabolite of disulfiram, which is a drug that's accepted for clinical use for prevention of alcoholism, and known as antabuse. And the active metabolite of disulfiram, DT-CMESO, has previously been shown to be effective at being protective in an epilepsy rodent model. We were interested in the fact that it's an NMDA receptor partial antagonist and it's neuroprotective in cell cultures. It's a selective carbamylating agent for sulfhydryl groups, and it'll prevent binding of glutamate to its receptors. Importantly, it's a partial NMDA receptor antagonist, as previous work has shown that total blockers of the glutamate system are actually detrimental. But partial NMDA receptor antagonists show promise, and a well-known example is memantine that has been in use for Alzheimer's, for instance. And this just shows that, in fact, we demonstrated that the DT-CMESO will carbamylate an NMDA receptor. In our stroke model, we've been interested in investigating a focal model of ischemic stroke, the middle cerebral artery occlusion model. And we hypothesized that glutamate receptor antagonists might decrease the size of the penumbra. This is a region that has a perfusion abnormality, that is a region that is capable of ultimately being protected given the right treatments, that can be recovered potentially, and where one sees neuronal paralysis but reversible ischemia. And then we have the core of the infarct, which we've also measured, where there's a diffusion abnormality and there's irreversible ischemia. So this was our model that we've employed for focal ischemia, and we used an intraluminal suture which was placed through the MCA for 120 minutes, and we measured blood flow using laser Doppler flow and maintained temperature control. So 3 different types of administration for DT-CMESO subcutaneously, either before reperfusion and then analyze it 4 days, continue for 4 days and look at later time points, 24 hours after reperfusion and test with 4-day administration, and then 24 hours after reperfusion and test for 8 days after reperfusion. Measuring lesion size is a standard method we employed, where we look at the volume of the control hemisphere and the volume of the non-infarcted tissue in the lesioned hemisphere, and VC minus VL over control hemisphere times 100 gives us a measurement of lesion size when we're staining with TTC stain. So the damaged area is the white area shown here. So we tested DT-CMESO on slices at multiple levels through the brain, 2 millimeter, 4 millimeter, 6 millimeter, and 8 millimeter shown here, and compared control with DT-CMESO treated. And you see in each case DT-CMESO shows a significant smaller infarction compared to untreated. And here we've done similar analysis. This time we're showing different administration, so 1 hour before reperfusion with DT-CMESO, or DT-CMESO administered 24 hours after reperfusion, and then analysis at 4 days and 8 days after reperfusion. And you see that with 1 hour before administration or 24 hours after reperfusion, in both cases there's protection at multiple depths through the brain. In some cases, the protection is only seen in 2, 4, and 6 millimeter if we use 24 hours after reperfusion. But nonetheless, with 24 hours after reperfusion, you do see protection at 2, 4, and 6 millimeters into the brain. And then here again you see protection if we look 8 days after reperfusion, particularly at 4, 6 millimeters. And in the case of administration 1 hour before reperfusion, we see protection at 4, 6, and 8 millimeters. So we wanted to analyze the signaling pathways, especially in terms of endoplasmic reticulum stress. And you see here a comparison of core versus penumbra. And we found an elevation in GRP78 mitochondrial. I'll explain later that GRP78, although it's an ER stress marker, it's also a chaperone. So depending on the subcellular compartment, it can play a pro-survival role. So what you see here is that with treatment with DTC-MESO, there's increased GRP78 mitochondrial in core and in penumbra, and increased GRP78 nuclear in penumbra, shown here. HSP27 is highly protective and plays a role in preventing apoptosis, as well as interacting with the cytoskeleton and preventing signaling to apoptotic processes. So HSP27, a small heat shock protein, is known to be a pro-survival protein, and here you see elevation both in the core of the infarct and in the penumbra for HSP27. Apoptotic components, BCL2 to Bax ratio highly elevated with pro-survival BCL2 predominating both in core and in penumbra. Again, BCL2 cytoplasmic is again elevated, and BCL2 mitochondrial is highly elevated. Cytochrome C in the cytoplasm is decreased, and cytochrome C release is known to be part of the pro-death process in apoptosis. It's decreased in both core and penumbra. And then cytochrome C mitochondrial, which is the source of the cytochrome C, is actually higher with DTC treatment. So that's a pro-survival aspect. BAC is pro-death, similar to BID and BIM, and you see that's decreased in the penumbra. And BAD likewise is decreased in core and penumbra. And then caspase-3, a pro-death protein, again is decreased in core, shown here, and in penumbra, shown on the right. When we look at the IRE1 pathway and the PERK pathway, PERK is an ER stress pathway, decreased function in both core and penumbra, and XBP1 is decreased in the penumbra. ATF4 is associated with the PERK pathway, and you see ATF4 is decreased here. PERK is already decreased, ATF4 is also decreased by treatment. Downstream, CHOP is decreased, BIM, pro-death, is decreased. This is a protein that's implicated in pro-death processes and DNA damage, and it's actually decreased in the penumbra, GAD34. And then PAKT, a well-known pro-survival protein that elicits a lot of protective processes, is highly increased in the penumbra. That's the region that is prone to recovery and can be saved by appropriate treatments. Just quickly to mention what I said about GRP78. Although it's a marker of ER stress, it does have pro-survival functions. And for example, in the mitochondrion, it can regulate mitochondrial energy balance. And in the ER, it's a chaperone activity, and therefore it can rescue damaged proteins, for example. So I'd like to turn now from our treatment that involved DTC-MESO alone to a multi-drug treatment. And I'll introduce two further drugs shortly. The approach, just a general protocol, again using a rat model of middle cerebral artery occlusion and blocking the blood supply for 90 minutes. We used 3 drugs, DTC and ASO, which I described, Sulindac, which we've investigated before in different model systems, including myocardial ischemia and also brain ischemia. Sulindac has been now established as a so-called, it's an anti-inflammatory drug. It's well known for a long time as an anti-inflammatory drug, but it actually protects, and we've shown that it protects not through its non-steroidal anti-inflammatory mechanisms, but through preconditioning mechanisms. And this has been demonstrated because the oxidized form of Sulindac, which is not an NSAID, also protects through preconditioning pathways, the same pathways as Sulindac itself. And indeed, the reduced form of Sulindac also protects through preconditioning. Third drug we're interested in is granulocyte colony stimulating factor, which is known to mobilize stem cells from the bone marrow and also potentially mobilize stem cells within the brain. It's also a neuroprotectant. It's been well studied as a growth factor that can elicit neuroprotective pathways in neuronal populations. And the approach we've used is either pre-treatment, 1 hour before surgery, Sulindac 2 days before, and GCSF 1 day before reperfusion, or post-treatment, where we administer the drugs 24 hours after reperfusion. And then we have subcutaneous administration every day for 4 days. This just shows Sulindac, which I mentioned, and wanted to emphasize that this has been shown to be a preconditioning agent, and by that it means it activates certain pathways that include mitochondrial functions that result in pro-survival responses. And some of the downstream targets of Sulindac can include HSP27, and in the heart it's been shown to activate INOS, and in the brain it can activate AKT, a pro-survival protein. And both Sulindac shown in the middle, the R and the S form, and Sulindac sulfone, the oxidized form, and sulfide, the reduced form, are all pro-survival. And then GCSF I just mentioned. Has been shown to be protective to neurons through activation of AKT and STAT pathways, as well as PIM1 kinase. And these receptors for GCSF are expressed in neurons. So it could both be protective and elicit stem cell mobilizing functions. So using multi-drug pre-treatment, we tested the levels of infarct, and we found that the pre-treatment resulted in a decreased infarct size at a range of depths through the brain. This shows pre-treatment with multi-drug after 4 days, and there's a decrease in infarct size shown here. Bax was decreased in the core of the infarct by pre-treatment. BCL2 is increased. And this summarizes some of the observations. So we have HSP27 was elevated as a pro-survival function, modifying caspase activation, and also playing a role in protection through cytoskeletal alterations. And also HSP27 is elevated with a decrease in ASK1 shown here. So again, we are interested to see if the ER stress pathways are affected by multi-drug treatment. We found GRP78 was decreased both in core and penumbra by pre-treatment with multi-drug. That IRE1 was decreased in core and penumbra. And to summarize the pre-treatment, we found that there was a decrease shown in red here in CHOP, JUNC, ASK1 and BAX, and an increase in BCL2 with the multi-drug pretreatment. We then tested post-treatment. We thought this would be particularly interesting as a potential treatment for stroke after a stroke's occurred. And again, we found a decrease in infarct size using TTC staining, shown here with post-administration. And GRP78 was decreased in the core significantly. BAX was decreased in the core. BCL2 increased in both core and penumbra, so the BCL2 to BAX ratio was in fact elevated in both core and penumbra, showing a pro-survival response. And HSP27 was also dramatically increased in core and penumbra. So we found that indeed there were increases in protection through decrease in key ER stress components. And in summary, with our findings, firstly in cell culture, we demonstrated that taurine and in vivo, we found that taurine can protect via ATF6 and IRE1 pathways, but not through the PERK pathway. DTC-MESO alone will protect via PERK and IRE1 pathways. And using a multi-drug strategy, in our pre-treatment approach, it caused decreased expression of apoptotic markers and protected through increased HSP27 and inhibiting the IRE1 ER stress pathway. With post-treatment, we've shown protection via GRP78, implicating the ER stress pathways, as well as an increased BCL2 to BAX ratio. So our future investigations may involve looking further at mechanisms of protection, perhaps also autophagy pathways, examining stem cell mobilization, especially since GCSF is part of the multi-drug treatment we've used, and looking at other combinations of drugs, including DTC, MESO with taurine. And this shows our funding. And then just to reiterate, this was the work of 3 groups, my own group, that of Dr. John Wu and Dr. Rui Tao at Florida Atlantic University. And these are our students and postdocs, Janet, Chandra, and Jigar, who were involved in the work. Thank you very much. Thank you. Are there any questions? Yes, thank you. We have a microphone.
Okay, my name is Hug from Vienna. I'm a vascular surgeon. I have 2 questions, sir. We have 22 amino acids with taurine. Why did you choose taurine? Okay, this is the first question.
Yeah.
And second, what I was a little bit missing in your concept is one enzyme system which connects us, endothelium and brain. It's a nitric oxide. We have an endothelial nitric oxide synthase, and you have neuronal synthase. And as we both know, nitric oxide is also a neurotransmitter and play, the role, of course, in neuroprotection. It's a neurotransmitter. So a little bit missing here, arginine or any cofactors playing role in synthesis of nitric oxide in the brain, during ischemia or, during the reperfusion. So could you comment
Absolutely.
a little bit on
thank
those
you.
2 topics? Okay.
Yes, thank you very much for the excellent questions. Our interest in taurine relates to its multiple levels of action. Taurine, plus its overall function in terms of cell survival. So it's consistently a pro-survival protein in multiple tissues. So it's pro-survival for kidney, For many ischemic disorders, it's highly protective. It's extremely effective in the heart against ischemic stress. And so in some respects, one might see that the pro-survival functions of taurine in the brain may parallel those in the heart, but not totally. And one of the areas it may not parallel completely may actually be in relation to nitric oxide, because the effects of nitric oxide and the pathways of nitric oxide are rather specialized in the brain compared to the heart. I don't have a simple answer as to the role of nitric oxide in the brain with respect to our studies, but what I would say is that both with individual drug treatments and multi-drug treatments, some of our treatments are most certainly modifying pathways that include nitric oxide responses. And one that I'm sure that applies to would be Sulindac. Sulindac's a preconditioning agent. Now, in the heart, it's straightforward. In a certain level, it's straightforward. It's a preconditioning agent that elevates levels of inducible nitric oxide synthase, and in a protective manner. In the brain, we've no evidence of it modifying inducible nitric oxide synthase, and indeed, if it did, that might be harmful. But that doesn't mean it doesn't affect nitric oxide function in the brain. I think there's already extensively reported studies on the role of nitric oxide in preconditioning in the brain. It's highly likely that Sulindac may be an example of an agent that activates some of those pro-survival aspects of nitric oxide, probably not through inducible nitric oxide synthase. In addition, I'd have to say that as we're modifying glutamate receptor mediated excitotoxicity through DTCMASO, and we're also modifying neuroprotective aspects through a growth factor, as well as potentially stem cell mobilization. All three of those functions must involve nitric oxide and nitric oxide signaling. So, although we haven't studied it specifically in our systems, it may well be that that's at the bottom of all the protection. And I would expect it's certainly a major player. Yeah. Thank you.
Thank you very much for a very nice talk. I have a question. You showed very nice protection here with decrease of the infarct size and apoptotic. But I mean, as we have seen in a lot of studies, it's working very nice in the preclinical studies, and then when we take it, it doesn't work. So I wonder how is about the functional outcome. Has that been studied in this combination drugs?
Right, thank you. Yes, thanks. Another excellent question. We have some data on a very primitive behavioral assay called the NeuroScore for some of our combinations, where we've shown behavioral enhancement relative to controls. But it's very primitive. It's not a very sophisticated behavioral assay, and there are much more sophisticated ones available. But in gross terms, the behavioral functions of the animals are better with the drug treatments. In terms of trying to stretch beyond preclinical into potential clinical avenues, I think we're very conscious of this as a important hurdle, and we haven't done this yet, so we can't really prove anything. But we have deliberately tried to choose a drug strategy that tackles the pathology at multiple levels. In this case, we have glutamate receptor partial antagonism that already shows promise in other clinical scenarios. In the case of memantine for Alzheimer's, which has shown some benefits. And that's similar to our DTCMASO partial antagonism without actually blocking important neurotransmitter functions of glutamate, and that's the big concern in stroke, is one wants to prevent the excitotoxicity but not prevent the neurotransmission, which is central to brain function. So DTC-MASO shows good potential for that, but also multi-levels of treatment may also show good potential. That's to say, a preconditioning agent that acts to a large extent through mitochondrial alterations, a glutamate receptor target, partial antagonism, and then we have preconditioning. And then we also have a growth factor that can enhance pro-survival signaling, but also mobilize stem cells, which may show, given an additional level of enhanced or conserved brain function, even more long-term. I think it's fair to say that some of the treatments that are tested in stroke may be influenced by the time of application as well as the effectiveness of the drug. So in a sense, the GCSF may show promise there because it's not only protective, perhaps short-term, but also stem cell enhancing more long-term. So we've tried to choose a combination that might be promising. All these drugs, it also has to be said, are already in use and approved clinically for one or other of certain applications. Each one has been through FDA approval for something, GCSF for various cancer applications and anti-inflammatory applications, and then DTC for as antabuse or the source of that. Disulfiram as antabuse, this is the metabolite. And Sulindax being used as anti-inflammatory. So at least the safety aspect of each of these has a certain track record. And I haven't mentioned anything on taurine plus DTC-MASO, but again, I would mention that taurine has a record of safety in a sense, and the combination with DTC-MASO might also be an interesting multi-drug approach. So I think in the context of needing new treatments for stroke, there's certainly a need for robust treatments for stroke. These may show potential. So I think so. Thank you. Okay. Thank you.